Let's see. Now I have to see if, there we go.
I just started the recording.
Okay. So should I introduce myself again for the recording or just keep right on going?
Sure, go ahead and introduce yourself. Sure.
Okay, so I'm Beth Johnson, Sisters District Botanist, and we have a rare fungi program for monitoring and managing rare fungi. And so I was asked to share a little bit about that and what folks can do to be involved with citizen science. So I'm the botanist, and I have a whole bunch of different parts to my job. One of them is dealing with invasive species, and that's a pretty huge portion of it, unfortunately, because that's such a pressing, critical ecological issue. We also have a pretty active native plant program where we collect seed, we have seed grown out, we reseed after certain disturbances. And then we have Forest Service projects, like so vegetation management projects, recreation projects, all sorts of things that I do the botanical analysis for and try to help design projects so that they benefit our rare species. Part of what I do is protecting and managing rare botanical species, and that includes fungi. So there's kind of a whole lot of different things that are part of this list, and that's one part. So I was pretty excited when Susan wanted me to talk and see if there's anything that folks that are part of this group could do to help would be great. So we have a rare fungi program, but there's really not a whole lot of us. So there's myself, a district botanist. There's other district botanists, although not all districts get to have a botanist. We have a forest botanist, and there's a regional botanist. We have a survey and manage and TES coordinator who is a really great resource for me. And then other than that, there's universities, there's contractors that we hire to do surveys. But there's really very, very few people who get paid to be mycologists. And that's why citizen science is really critical to a lot of the knowledge that we have. So I say we manage rare species, and what is rarity? So there's different ways that something can be rare, but basically it's something that's either it could be very common in a very small area. So I have a picture here of one of our vascular plants. It's a Pex penstemon, and it only grows around the Sisters area, nowhere else in the world. But around the Sisters area, it's actually really common in the ponderosa pine. But because it only grows in that area, we have to be pretty conscious of our responsibility to protect it. We also have some rare species that are really widespread. So some of our fungal species are found in Europe, they're found all over the place, but very, very few sites for any of them. So they might have a really wide range, but very, very few sites. And then some species were just right on the edge of their range. So they might be a species that's common in one place, but we're on an edge of their range for where they exist, so they're rare for where we are. And we want to protect those too, because those edges can really have some unique genetic variation there. So why are species rare? Some species are inherently rare, that they've never been very common. And then there's also things that we've done that have had a huge impact, that whole sixth extinction event that there's a lot of talk about. We're part of that, or we're really driving that. So things can be rare because they have really unique traits, or they can be really adapted for a really small, very specific habitat. Some of them have really slow life cycles, so they don't reproduce quickly or frequently. And then threats from humans. And some of our threats include climate change, invasive species, habitat conversion, uncharacteristic wildfires can be an issue. And then there's some other considerations, and one is just detection. So for some of our rare fungi, like they might be more common than we know, and we just haven't detected them. Or nobody's really capable of identifying them. Because there is a pretty small group of people with the skills to identify some of these things that require a microscope, chemicals, so it's pretty hard it's not something that a lot of people would notice. So why do I care about rare species? And for a lot of us, I think we recognize that things have intrinsic value, that it's the ultimate in human hubris to think that we have the right to make something go extinct. I think most of us here probably recognize just the intrinsic value of every species, but not everybody does. So there's some other reasons too. So one is that we don't understand the cascading effects of what a loss could be. Ecology is incredibly complex, and the loss of a single species can have a lot of effects that we're not able to predict. So we don't necessarily know what's going to happen when we lose a rare species. They can be a sign that an ecosystem is really falling apart, and we as humans depend on these ecosystems for ecosystem services of water, clean air. And when we have a system that's falling apart, that could ultimately affect all of us. The next is that in one study they found that rare species can have an outsized effect on the rest of the ecosystem. So you have one species that can have a much larger effect, even though it's rare, than something that's common sometimes. And sometimes those effects are different. We can't necessarily predict them. They can be, so these rare species, some of them are helpful for how the systems can respond to changes. And then on a really more selfish level, we can find cures for diseases. A lot of our cures have come from plants and from fungi. And also wild relatives of some of our common crops have important genetic material that we can use. So there's a whole bunch of different reasons that we are trying to protect our rare species. For fungi, we probably have only identified about 5% of the world's fungi, so we don't even know what we have. And one of the things for surveying is that the more information we have about what's out there, is the better job we can do for protecting things. So as a Forest Service employee, there's a few, so there's the big reasons, and then there's also some legal reasons. And so the first is that we have the Endangered Species Act that actively protects endangered species, and it's a legal mandate. On that species, that list right now, there's only 2 lichens, and so those are the closest we come to a fungus, because those are really a symbiotic relationship between a fungus and an alga. But we have no other individually listed fungal species. We also have survey and manage, and I'll go into that. That's a big part of us living here in the Pacific Northwest, is the Northwest Forest Plan and survey and manage protections. And then we have a sensitive plant program that's part of Oregon and Washington that includes all different habitats. So the history of protection was in 1994, the Northwest Forest Plan was implemented, and it was established to protect the habitat of the northern spotted owl and associated species. And there was a recognition that, first off, the owl's primary prey items eat a whole lot of fungi. And then second off, the fact that the owl is endangered says that the rest of the habitat, a whole lot of other species who are dependent on it could be as well. So they used a whole bunch of different mycological experts. A whole bunch of people came together and came up with a list of 234 different species. Many of them are fungi, but not all of them, that we would survey for and manage. Since then, that list has actually been reduced because they found some of those are more common. Once we started actually looking for them, they weren't quite as rare as we thought. And I'm going to go through some of the survey and managed species that we have on Sisters District and on the Deschutes National Forest. And then there's also different categories. I'll talk about mostly the category B ones that we have, but then there's also some other categories. I won't go into too much detail about that though. So following after we had protections for fungi with the Northwest Forest Plan, there's also Region 6. Actually, every region of the Forest Service has a sensitive species program, and ours is Region 6, Oregon and Washington. And the goal of this is to prevent species becoming threatened or endangered. So we don't want them to have to get listed. And so the list that we have for Region 6 is species that if we are careful now, we can prevent them from getting onto the federal list. Every region has its own list, and it includes all different types of habitats. So it can be habitats like vernal pools. It can be, sometimes it's very disturbed habitat, sometimes it's old growth. It's any type of habitat within Region 6. So for me, I have the support of the ISSSSP program. And this is an interagency program. So as I said, like I'm a botanist and fungi is a very small part of what I do, but this is a group that is a huge resource. And they also have some really cool resources for the public too that are on their website. So they basically follow the policy, but they have folks that have helped identify what our high priority species are and what those habitats are. They help look for data gaps and information needs. They've done conservation assessments and strategies. So there's somebody that I work with, and you know, if the group is interested in more in other kind of a better picture of the rare stuff in particular, this would be a good group to go to for another speaker at some point. So some of the things they've done is that they've done random grid surveys like all over the Pacific Northwest. They've come up with some models for what kind of species might occur where, and then habitat recommendations for what little we know sometimes for these rare species of, you know, can they handle prescribed fire? Is that going to be beneficial or harmful? What about understory thinning? Is that going to be beneficial or harmful? Because not all species do best without any disturbance. There's some species that actually require some disturbance or they disappear. So trying to identify what is best for each species and then helping provide me with that information for when we have a Forest Service project that we can try to adhere to that and really try to enhance habitat to the extent possible.
So maybe if you move back from your microphone, it won't be quite so boomy. I'm not quite sure. It's really loud and kind of boomy.
Okay, let's see. I don't know if I can, so I'll sit back here. Is that better?
Well,
No.
probably not. It just, I don't know, maybe it's just somehow the mic is kind of being really loud and kind of rattly. I don't know, maybe other people are having that trouble too.
Okay, do you think I'm talking too loud or is it just my microphone? Because I don't know what I can do about that.
I know, I think it was just, go ahead and do what you were doing before. It's probably just as well. Either way, it didn't seem to matter.
Okay, I'm sorry. So this is a cool thing, like if we're all stuck inside this weekend with lots of smoke, I recommend checking out a couple of the websites that the ISSSSP, so 4S's program has, and there's links on there to all sorts of different things. So there's handbooks and for identification, there's species descriptions, there's photos, there's so much stuff on there. You could spend weeks on the website if it was really smoky for too long, which hopefully it won't be.
Can you tell me who is this organization through? Is this a government organization?
It is. It's primarily Forest Service and BLM. So it's an interagency group, but it partners with a lot of other organizations as well, including universities. So it's primarily through the Forest Service and BLM, but with a whole lot of partners. And so it's a really cool organization. I don't think they have very much funding like so many things. So they do rely on, you know, some citizen science as well. The BLM has their own website that's separate from it too, that has some good stuff on there as well, like some of the ID stuff. But really, like I can spend hours and hours on the Interagency Special Status Sensitive Species Program website, just looking at all the different resources they have. Some really cool, like free downloads and stuff for guidebooks and all sorts of other things.
They have a good one on fungi?
They have a really good one for rare fungi. And so I'll show you that one in a second. It's a little hard to use because it's a key, but it doesn't, it only includes the rare ones. So you have to key it out precisely. And it's not one of those ones where you can say, oh, it's not that, so it has to be this. You have to really get all of the characteristics in particular right. So it is a little challenging to use, but it is really good for the rare ones. And this is a screenshot of it. so it has stuff on all sorts of different types of organisms, and a ton of it is about fungi. So these are some of the things that you can download from there that are pretty helpful. Species fact sheets, information about, for me, like buffers for how to protect sites, conservation assessments. For me, they can also help with some specimen ID and verification. And then they do have some other field guides. So it's definitely worth checking out if you haven't seen it before. Separate on the Forest Service, there's some other government publications that some of you may or may not have seen. These are all free downloads also on the internet. So books, so guidebooks specifically on Romaria, on the ecology of morels, on truffles. So these are separate from that rare website though. So Susan asked me to talk about how the group could be involved, and so there's three main different ways of if folks in the group are interested in rare fungi, what they could get involved with doing. And one is that I have a bunch of sites that are historically known sites that some of them nobody's visited in 50 or more years, others 10 years, and you could adopt a site. Another one is learning one or more of the rare fungi and just keeping an eye out while you're out. The other is some groups have done complete species lists for areas as a group. So the first thing oops, I probably should have put this in a different order, but these two handbooks are they're where I got a lot of my information and a lot of my photos and basics for the slides. And these are free downloads. They're a little older, unfortunately, so 2003 and 1990 something or other. They have some updates in the website that you have to click to individually, but these have some great pictures, all sorts of really great information. So I do recommend downloading those if you're interested in rare fungi.
I have those in hard copy, and what I found really interesting are the maps where they have the map of Oregon, and then they show exactly where they're being known to fruit. So you can go to those sensitive species areas and oh, you know where they're hunting because you've been in that area before. So I'm like, well, that's where they found blah, blah, blah. And it's kind of fun just to see in the areas that we hunt, which maps correlate to the places we've taken field trips and stuff. Because there are some that correlate with those spots.
Yeah, and so the books are older, but in the website, there's also links to updated maps for a lot of the species that are more recent. So they include even more sites if you get really into looking at those maps. I definitely recommend checking out the updated versions, although some of those are still, 7 or 8 years old, but at least they're, they're not 20 years old. So the book, so I'm going to talk now, I'm going to start talking about some of the individual species that we have on Sisters District. And the book breaks them up into taxa groups. And so that's how I'll go through it too, is kind of lumping a few different groups of species together. I included a web link on here for something that has some really good pictorial keys, and it also talks really well about ID. So I don't know, it's it's a .com website, but I thought I think it's really helpful, especially when it talks about some of the microscopy work for identification, and also some of the chemicals and stuff. So the first group that I'll talk a little bit about is our sequestrate fungi. And these are ones that have to either be eaten or decompose in order to release their spores. And this is a list of the ones that we know or suspect on the Deschutes National Forest. So you can see there's quite a few that, for Sisters, that we know we have at least one or more sites of these. And so I'm going to talk about a couple of these, trying to focus on the ones that are easier to identify. Because like I said, there's a few species that really, like identification, requires chemicals and a microscope, but then there's also some that are fairly easy to recognize and that we need to do some more tests to confirm them, but they're actually not that hard to ID initially. So I'll be focusing mostly on those. For in the book, they have description forms for each taxa group. And this I find really helpful because it tells you what some of the most important features are to look for. So shape, whoops. Ooh, that's our first one. So the shape of the mushroom, I guess. Color upon collection, color after bruising, texture, color changes with chemicals, arrangement of the gleba, which are the inner portions. So these are just things that you would look at if you were looking at a specific individual for identification. And I think it's kind of helpful sometimes to know what characteristics you're supposed to look at. And something that's always important is the substrate. You know, especially if you collect something, it can be really hard to remember what it was growing on. You know, and even for some of the species, whether or not it's growing on a conifer or a hardwood can help you identify which species it might be, even if they look almost identical. So this one is really cool looking, although I've never seen it in person. There's only 3 known sites in Oregon. There's also a site in Idaho, and then there's some in Europe. But it is found in the Metolius Basin in ponderosa pine. And really, we have 2 of the only known sites in Oregon. You can see on the map, this is the map that Linda was talking about. And can you guys see my pointer? Yeah, okay. So you can see we have 2 sites really close together in the Metolius Basin, and then there's 1 site over on the coast, and that's it for known sites for this species. The ones on the Metolius were last found in 1985. So who knows if it's still there. It's, of course, even if we couldn't find it for a long, long time, it's very likely it's still there and just not fruiting. But this one is distinctive in a couple ways. One is that this is the peridium, the white part, and it isn't always white, but it's really thick. Or sorry, the black part along the edge of it. So it's really thick. It's at least 3 millimeters thick, and that's an identifying factor. The outside is warty and black. So those are the main identifying characters. So they are pretty distinctive overall. So we have 2 known sites to that. That I'd love to know if they're bigger. Maybe the sites are bigger than we know. We really just have 1 single point from 1 individual at each site that was found. The next species, we don't know that if we have this on sisters, but this was found just a year ago for the first time ever on the Deschutes National Forest. And I thought I'd throw this in because it's a more distinctive one. So Gastroboletus vividus, and the vividus part, it's really vivid yellow and orange coloration when fresh with orange and red. It looks like an aborted bolete. It's usually partially buried. There's no glandular dots on the stem, and it stains red, not blue. So this is one that is also somewhat distinctive, and it was found just like a year ago off of the Cascade Lakes Highway near a major trail. So it's obviously here, and just nobody's seen it for some reason. Who knows why? So for the map doesn't even show our Deschutes site because it hadn't been found here when it was found on the Deschutes. So the map was made before it was found on the Deschutes. It's not known on sisters at all, but I sure would love to know if it's around. It very well could be, and it would be great to be able to expand what we know about its habitat. We do know that it's typically associated with moist old growth forests, the moister ones, so not like the drier ponderosa pines. It's believed to have ectomycorrhizal association, so it has a symbiotic relationship with trees, particularly Shasta red fir, Pacific silver fir, Doug fir, and hemlocks. And this is one that's on multiple groups, so it's protected as a survey and managed species and also as a sensitive plant species. So this is definitely a higher priority species as a result. We have 5 other Gastroboletus species that we could have on, that we have on the Deschutes National Forest, and some of them are much harder to identify than others. We do know we have some sites for Gastroboletus ruber up near Carl Lake, and we have a few different cool rare fungi sites up near Carl Lake, which is on the Mount Jefferson Wilderness. We also have Gastroboletus turbinatus, and these can be harder to identify. So they're definitely something that would be a more, intermediate to advanced level thing to be looking at. Another one also on Sisters that we have just one known site of is Rhizopogon alexsmithii. It's commonly called a false truffle. This one's associated with hemlocks. It has, it's kind of pinkish usually, sticky with pale marbling, and it's associated usually with hemlocks. And we have, I think, just one, let's see, we have 4 sites on the Deschutes National Forest, and I think just one of those, I believe, is on the Sisters District. This is another one. It'd be great to, be able to determine if it's, where exactly it is. We have one known site. Is that site bigger than we think? Is it smaller? How often does it fruit? It'd be neat to be able to go back regularly. And unfortunately, I would like to do that. I haven't really been able to, though. This is another one. This one's really hard to identify, but it does, but we have a couple sites of this. Would also be great to have somebody go and check out for us. Archangelialia lacteroides, and I apologize if I pronounce things wrong. I think for a lot of botanical terms, we just try to say them with a little confidence, and I've heard a lot of people say a lot of things very differently. And so I apologize if I don't say it the way you like it to say it. Most of these are things that I read and don't hear very often. So we have one site of this up on Green Ridge that was found in 1999, and then another site that has an unknown date. It just says 1900 in our database, which I doubt it really was 1900. Up near Jack Lake. And that's why I included these, even though they're pretty hard to identify. They do have kind of a whitish fluid that comes out of them when they're broken.
Is that last one, is that a buried? Are these hypogees, are these underground?
Yeah, all of these are ones that are either partially or fully underground. So we only will find ones that, some of these can come up partially, but they're all partially buried at least. So we'll find ones that have been dug up by rodents or other animals, or some of these come up just a little bit, just enough to see them. Which unfortunately quite a few of our sensitive and rare ones are like that because they're really hard to find.
And that's because of Dr. Trappy from University of Oregon, I think.
What about him?
OSU, and he was, really into truffles and hypogeous fungi, and, he's kind of world renowned for that. So he was out in the early days looking for those. And of course, with his focus on truffles, that they're all going to be weird stuff and hard to find.
Yeah, quite a few of ours, like his is the name that's attached with the person who found them. So yeah, he was the one who found a lot of these sites on Sisters District. So our next group is the polypore fungi, and these are ones that the underside of the sporocarp is either tubular or porous. And so for this, we have a handful of different species. A lot of these are pretty hard to ID, and a number of these have only been found further on the west side. So we don't really think they're likely to be found on the Deschutes National Forest just because we're a little bit drier. But for that, some things that we're really looking for include flesh consistency, whether it's fleshy, brittle, or spongy. Surface ornamentation can be really important, and then surface texture. Is it dry, sticky, greasy? For the stem, what shape is the stem? The stem can have a different texture than the cap, and so that can also be important for identification. So there's quite a few different factors to look at for that. Analysts, so they're kind of like basically the little ring around it, whether or not there is one, if there is, what color it is. And these are actually two really cool ones that we don't know of on sisters, but so there's two species. There's this one first, and then Flettii. So Albatrellus ceruleoporus, and it's one that's pretty distinct. So this is one that would be pretty easy for people to keep an eye out for while they're out and about. So it grows on soil, so that's its medium. So it's not going to be coming out of wood typically, but out of soil. It has spines on the underside. The cap and pores are indigo blue to blue gray. And then we also do talk about spore size, but in general, if you find something that looks like this, we would confirm it with spore size, but it'd be pretty likely to be the Albatrellus ceruleoporus.
What habitat for it?
What's that?
What kind of area does it grow in?
So it's so it has been found on the Deschutes National Forest up in the High Lakes area, in the wetter kind of in wetter habitat up near the High Lakes. So we have the Albatrellus, this Albatrellus, and then the other Albatrellus looks quite different from it. If you're looking online, there are some debates as to what species grows where. So it's a little bit confusing there, but we go with those two handbucks. And this one does not have the spines, instead it has pores. It also has that really distinct blue color though. There's some that looks more similar to this than to the other one. But this is much more blue than any of the other species. So another fun one to keep an eye out for while you're out mushrooming for other things.
I think we find that a lot.
Mm-hmm.
So it's possible, so that would be one that if you found it on Sisters District, I'd be really thrilled if you brought a sample to me and then we could see and confirm whether or not it actually is, because there are some others that could look pretty similar. So I'd want to make sure with a bunch of different factors. But it's possible that we do have more than we realize, and that would be really cool to know. So a few other Basidomycetes, and again, I'm going to try to focus on ones that are easier. I'm just going to focus on one in this case that's easier to identify. Again, for data collection, there's a bunch of different things you look at specific for guild fungi. So for this, texture is again really important. So is the substrate that it's growing on, the stem consistency, flesh texture, then gill characteristics, how they attach to the stem, whether or not there's a veil, width, size, shape, those things can be really important. The color can be important. And then also for some of these, we do chemical tests. And so there's two main chemical tests that can be done that are the primary tests done for many of these species. And some species can be identified without them and others cannot. But basically you put the chemical on the mushroom and see what color it turns. So this one, I think, so we have a couple of sites down in the Metolius area, and I think this one could be more common than we know in the Metolius, I hope. It's found only in mountainous regions of Western North America. It's often found near melting snowbanks, and you can see the color for this is also a pretty distinct factor. So it has this kind of sickly bluish color to it. We know that it fruits in May and July, but it could fruit at other times too. And that can be another important factor for what time you find the fruits. And it seems to have a mycorrhizal relationship with some of the pine and, so pine ABs so pines, firs, hemlocks, other pine relatives.
We found I found this with a botanist over by Jack Lake, and they had a Jack Creek, all of that up there. They found that hygrophorus there, and it was definitely green blue, and they were very excited about it. It was like as you're going into like the head of Jack Creek was on the right side of the road going in when most of us get up there, you know, during morel season. So if you happen to be in that Jack Lake, look for this really cool waxy cap. Hygrophorus is a waxy cap. And this is, so it's got pretty thick gills, but it's so distinctive because of the green-blue gills underneath. So it kind of stands out when you take the time to look for it.
I'm guessing that probably the site that you found is probably the one that, one of the sites that I have listed as a place that we'd like to have revisited, I'm guessing. Do you know how long ago it was that you were up there, Linda?
Say that again?
How long ago was it that you were up there with that botanist, do you think?
It was quite a long time ago. I don't know, within, you know, probably more than 5 years. But something like that. I wish I could remember. But
well, that's cool. That's really cool that you were at the site. That's really fun you were at the site, because that's one of the sites that'd be great to have somebody revisit and see if we can expand, you know, the location, you know, where we know it is or not. So one thing that I've read, and I'm not familiar with though, is apparently it's supposed to smell like rotten cornmeal. Did you notice that smell when you were seeing it?
I don't remember because I didn't know it. So that do it. So I'm like, oh, so yeah, I think that it's, yeah, that would be really cool to know, the smell. Rotten cornmeal? I mean, how does rotten cornmeal smell? I don't even know.
I don't know. I think we're going to have to go smell one of these to find out. Yeah, apparently that there are a couple other species that look somewhat similar, and that rotten cornmeal smell is one of the diagnostic features to make sure it really is this one, and not a somewhat similar species. So you can see that on the map, these are our sites up there in that Metolius Basin and up towards Jack Creek. And then other than that, you know, a lot of the sites are a lot further south or a lot further north of us. So this group, I'm not going to go into any detail on. So this is one of the most challenging groups, in my opinion. Linda, what do you think? Is this one of the most challenging groups for identification?
Well, I went to Ron's workshop, who wrote those Ramaria for the Pacific Northwest books. It was a 2-day workshop, and by the end of the first day, I was like over my head. I was so overwhelmed that I couldn't cope. But I'm not a mycologist either, and you know, I'm not as used to all the microscopic work that it takes to figure out Ramaria. But there are some very distinctive Ramarias, but for the most part, they're really hard because they change from when they're little tiny to when they're big. They completely change color, shape. You know, it's, to me, you have to, I don't know about people who like Ramaria study, but I'm like, wow, that's a different kind of mentality than I have. So no, I find it very, very challenging, although I do happen to like a couple of them to eat. So at one point you finally go, well, it's close enough. You know, it's close to that one if it isn't exact. So, and I don't know anyone that's been really seriously hurt by eating Ramaria. If that were the case, we'd know about it by now, because some people just go out them indiscriminately and pick any big Ramaria and eat it. At least I know people who have done that, and it hasn't been a problem. So finding, I know you're going to say something about trying to find some of these. Good luck. You got to be a real Ramaria geek person, you know.
Yeah, this is one that I'm not saying, oh, hey, people keep an eye out for these. This is really, like, you know, if there's somebody who's part of this group who's really into really hard to identify obscure fungi, then this is the group for you. And I decided to bring it up because we have quite a few of them. They're super hard to identify, so they probably are way under recognized. And our botanist on Crescent District, Christina Vaverka, she is really good with them. And so she's actually found quite a few, and she's been able to actually find enough to have some of them, you know, one of them removed from the sensitive species list because it really wasn't that rare. It just wasn't identifiable. which is pretty exciting. You know, it's really neat when we find something is more common than we thought it was. So I'm bringing this up just because we do have a few different sites of this. But for these, you know, there's a lot of different things that people look at for identification factors. You know, the stem form, flesh consistency, branch consistency, odor, width, and then really those chemical tests and spore size and shape are really critical to these. So yeah, we won't talk about these for very long. But here is one that we know we have on sisters. But you do have to do the, you do have to see what, if it has a reaction to the Melzer's reagent. You look for a rusty root to it, and then you also have to look at the spores, and they're short, narrow, and cylindrical. And then some other species of Romeria have one or more of these characteristics, but not all of them, including the fruiting in September, October. That's another thing that's important, is the time of year that this one fruits. So that's it for the Romerias for me. I thought about having one other that we have in there that keys out pretty easily in the Mushrooms Demystified, but it keys out to it and others, and the and others part is kind of where it gets pretty confusing. So the next group is another group that there's a few that are more distinctive that we know we have on sisters. And for these, again, we're looking at, as always, looking at substrate. For this, looking at the shape, stem, flesh color. The texture is really important. There's some of these that can be almost hairy, others that are really smooth on the outer surface. So there's a bunch of different features that we're looking at just from how things curve. And then, as always, you know, for some of the more detailed, you know, we are looking at the spores as well. But you can see for here, sporocarp shape. So the top part of the mushroom, you know, is it a cup, a disc, cushion, a rabbit ear? You know, so there's a bunch of different ways they describe the shapes. And some of them I'm not totally sure what they mean exactly by their description, quite honestly. So this is one that we have on sisters, is the Helvella crescitunicata, or elephant saddle. There are a few similar lookalikes. But there's a couple of distinctive features with this, and one is the time that it fruits. And there's a lookalike that fruits in the spring that is not our sensitive and survey and managed species. But if it's fruiting in the late summer and fall, the cap is cup-shaped and rubbery with really deep rounded ribs. And basically the way the edges curve is supposed to be pretty diagnostic. And for this, we have two known sites. One's up near Shirley Lake, which is is right about here. It's kind of near Carl Lake, and the other is near Three Creeks Lake. So two sites from this one. It's, you know, definitely like an intermediate to advanced one. It's not super simple to identify. There's quite a few similar lookalikes, but if you really get down into some of the details, then it is usually macroscopically identifiable. And then another cool looking one, and this also has, unfortunately, a lookalike with different spores, but there's are a couple of pretty distinct features, then it can be identified without the spores, at least initially. And one is that, so it's called the umbrella false morel. It has really deep stems. You can see how the stems are really deep and widely spaced. The cap is really thin and spreading, and that's why it gets called the umbrella false morel. The stalk is often kind of reddish or purplish tinged near the base. This one, we have only one known site on sisters, and that's up on the way into Three Creeks Lake area before you get there near Black Springs and Black Springs Campground. And last time it was found was 1991. You know, it'd be interesting to know if it's still around. There is one other, like I said, similar species, but in more recent years, they've been able to differentiate the two, let's see, do I have it, without using spores. Next group, and this is another one, I'm not going to go into I'm going to talk very briefly about one of these, and that's about it. But the club fungi and others is how they break it up in the handbook. For this, we're, as always, looking at substrate, width of cap, odor, taste, don't swallow, flesh color, length, and then surface characteristics.
Can you tell me when does that umbrella of when time of year does it fruit? Is it a spring fruiting or a fall fruiting?
I'm gonna have to re-look that up. I think it's fall fruiting, but I actually have to look that up to answer that, sorry. Let's see, I think I wrote it down to try to remember. Let's see, I probably have it handy even. No, I did not write that down, so I'm not sure offhand.
We can look it up later because I'm recording this, so we, if somebody would like to know, because I do think we find this. It seems like in the spring is my thinking that we found it. So I'm not sure where, but like you said, there might be a look-alike that looks a lot like it, but I know I've seen it before.
Oh, cool. Yeah, it's a really cool looking one. So the club fungi, so this is another one that's really hard for anybody to survey for. Christina Viverka, the botanist on Crescent's been able to find some. This is one that Mushrooms Demystified, you can key to it, but then it says and others. And really you have to look at the spores as the final determining factor. And so even though this is another group, kind of like the corals, it's like, well, if somebody wants to look for them, we would love it, but it takes a really high level of skill that very few people have. But we do have one site that's kind of near Highway 242, the Mackenzie Pass. We have several other, two other known species, but both are hard to ID. Okay, so the last three are fun ones that people can eat. So the blue chanterelle, I love this one. It's very distinctive. It's blue to black. It looks like a chanterelle, even though it's a different genus with those shallow crowded veins or wrinkles, not true gills. It's fan or spoon shaped, not a horn or tube. So that's important because there are some things that look similar that are horns or tubes that are not the blue chanterelle. It's found all over the place in China, Japan, Korea, Maine, Oregon, but it's not that common. But you can see it's, of our rare species, it's more common than many of our other rare species. It's often found with spruces, firs, or aspens. And this is a one that if you're very confident in your identification, I'd say it's delicious. Linda, do you have any comments about this one?
Other than that, it's one of my very favorites, and when I found out that it was on the sensitive species, I was really surprised because we find it pretty common I wouldn't say common, but I don't know if a fall goes by when it's not been found by the group or even some of us. And of course, most of us aren't going to say because, we don't want to tell people where it is, but
yeah.
Is that something you're saying on their Sisters Ranger district? I found it certainly over here up near the High Lakes. And I'm not sure how far west your district goes, where Willamette starts, that's kind of a gray area in there about when does the Sisters district end? Because on that side, we have found it too on field trips.
Yeah, we end kind of right near the ridge line there, like so right below right near the the Pacific crust basically. So we have everything that's east, and so this one I think is a little more common west. And yeah, I haven't seen it anywhere on Sisters District. I have seen it on Bend, Fort Rock, up in the High Lakes area. And yeah, it is one that, I don't know if people want to share when they find it or not, but it's just fun for, I think, folks to know that it's, that it's one that's being managed for. So this one is on the survey and managed list, not the sensitive list. It's mostly because it's associated typically with with old growth trees, in my understanding. Now there's a different category of survey and manage, that's survey and manage category D, and this is one that we don't actively have surveys legally required, but if we have known sites, we manage for them. So I threw this one in. So it's it's not that uncommon, but it is on the survey and manage list because it's typically associated with old growth. But this is the Spiraceus crispa. There's some debate on names depending on where you go. There's some name, what species it should be called. And I go with what the survey and manage list says, which is the crispa. But it's really the only species we have of Spiraceus here. So if you want to call it the other name, that's fine. But it looks kind of like a cauliflower, really ribbon-like, and it's another popular edible species.
I was understanding that this one is a parasite eventually, that it is a very, very slow-growing parasite on old growth. But it takes many years. So, I mean, it's never been a concern. I think that, I could be wrong, but I seem to remember that because it's such a delicious mushroom. And, I have seen them monstrous sizes, maybe 3 feet across. And by then, usually the worms have set in. And so it can get very huge in a good year with a lot of water. But I was kind of dismayed when I found out that this delicious, beautiful mushroom is not necessarily a good thing to find. So I'm not sure. Do you know about that?
I have not heard that. It's possible. I would have to do some more reading. My thought is with any of our native parasites that it's an important part of the cycle of life anyway. So, it wouldn't bother me too much. But yeah, I don't know that. I'd have to look that up. Thanks. Then our very last species is not survey and managed in Oregon, just in Washington and California, but it's the white chanterelle, and it's always fun to see pictures of white chanterelle. So if you were in Washington or California, then that's on the survey and managed category D list because it's less common. And we're pretty fortunate here in Oregon that we have quite a lot of it. And this year, I was even seeing it right next to the Metolius River this last fall, where I hadn't seen it before. It was just such a good year for fruiting for things. All right, so to go back, and so some of you are probably like, okay, that's great, have fun with that. Now I'm going to go back to collecting edibles, and that's fine. For folks who are getting excited about doing some kind of citizen science, I'll go over, this is the same slide as from before. I'll go over, I won't take too much longer, but those 3 main ways, which are revisiting sites, looking for rare species, or doing a big group. So for a site visit, if somebody's interested in that, I could give them a map and species info. The person would probably want to do, some more research before revisiting the sites, know as much as possible about the species, and then try to revisit the sites a few times at different times of year, at different times through the season. If somebody wants to do this and they find one, I'd want them to collect a voucher specimen. I'll talk a little bit in a minute about how we collect voucher specimens. We need to have GPS points and documentation with the forms filled out. So if somebody wants to do this, it's not like just go and visit the site and have fun. It would be, actually go and try to do something. Otherwise, we tend not to give out our rare species sites very much. There's worries of what people might do. So we usually only give out site information to people who, seem like they really have an active interest in volunteering. But we have 44, on Sisters, I have 44 different sites with 23 different species. Some of them are really hard to ID, and so probably nobody would want them. And then others are pretty easy to ID and might be, a fun hobby for somebody. The next are target species searches. And for this, you saw a bunch of different species. There might be ones of those that you start seeing out that you want to record. There's a couple of different ways to do it. If it's for me and we're really certain it is, then we want to collect a voucher specimen and go through a whole process with the region to document it. There's also ways to do it a little more casually. So a really great group is the Fungal Diversity Survey group, and I have their website listed down here. It's a way that people, instead of volunteering directly for the Forest Service, they go out and do some documentation on their own and work with this group, usually documenting things either on mushroomobserver.org, which is awesome, or sometimes on iNaturalist. But the Fungal Diversity Survey group, they also do some genetic testing. They kind of have some higher level ability if you want to get involved with that group. I definitely recommend checking it out if that's something you're interested in. And then the last is like the full survey thing. And in the past, there have been groups that have done this elsewhere in the region. Our forest botanist, Jennifer Farrell, she's worked with groups like on the Blue Mountains where they've had volunteer groups come out and they all work together and survey a whole area together. But usually that takes a lot of organization. So that's not really something that I'm pushing for or anything like that. But it's just, it's something that if the Mushroom Club was interested in, that somebody could try to instigate as a fun group activity. For anything that you do, kind of a lot of the similar equipment that you would use on your own for collection, except also each individual needs to be wrapped individually, and I'll explain why in a second. You need to have field tags and data sheets, a GPS, but the smartphones work great for that, pens, and then your books. So mostly what you normally would have with you. But things to consider before collecting. One is if you are revisiting a known site or you're finding one, is to avoid excessive disturbance of the site. And so that's one of the reasons we don't give out our locations very much, is we don't want to have people disturbing a site heavily. The next is that each individually typically needs to get wrapped individually. And the reason for that is that the spores can get mixed together. And for really confident ID, you have to be able to identify it through spores. And if the spores are all mixed together because everything got kind of stirred together in your basket, that can be really hard to do. We also usually try to keep them just moist enough until you can get a spore print for ones that you're doing a spore print from. And then we also do clean tools regularly, because even just the cutting them, you can get spores and other stray particles on between samples. And then the form, so a substrate, super critical to know. And then the last thing is to say, even the professional mycologists make mistakes, both in the field and in the lab. So not expecting perfection. So pretty much I'm wrapping up now. Here's some websites for more information. I do really recommend checking out this fundus.org in particular if you're interested, and then mushroomobserver.org. I got a whole bunch of my photos from them. And some of the photos could maybe not be actually what I think they are. It's a website that people put their photos on and do group identification. But the ones I chose seem to be right. And they do have different levels where they have folks who confirm. They have a whole bunch of different levels for it, but people put things on there that they just say fungi. And then they also have people who put things on there with very specific identifications. And it's kind of a group process with some really good experts who put a lot of time in on there. So I recommend checking those things out. And that's it for this talk. Any